Yeah. Ok.
Então, vamos lá. Então, boa noite a todos, bem-vindo novamente à nossa reunião semanal do Centro de Doenças da Mama. Hoje a nossa reunião vai ser coordenada pela patologia, pela doutora Ana Paula Sebastião, e nós temos uma convidada hoje, a doutora Fresia, que é patologista do Memorial Sloan Kettering. Good evening, Fresia. Welcome to our weekly breast cancer meeting. It's a pleasure to receive you in our group to discuss this very important topic, okay?
It's my pleasure. Thank you so much.
Yeah. I will pass the coordination of this meeting to our pathologist here, Dr. Ana Paula. Please, Ana.
Okay, thank you, Karina. Well, it's a great pleasure to say that we are resuming our meetings from the Centro de Doenças da Mama with an outstanding international speaker, my friend, Dr. Fresia Pareja. In addition of being a medical assistant as breast pathologist at Memorial Sloan Kettering, Dr. Pareja also is a researcher at Dr. Jorge Reis Filho Lab, involved in the cutting edge of molecular breast tumor research. She published more than 100 peer-reviewed papers till now in this subject. The title of her talk will be Genotypic Phenotypic Correlations in Breast Tumors, New Diagnostic Applications. So, Fresia, we are really excited to be hearing about breast genomic pathology, applicability, and correlations. So it's a great pleasure to have you here tonight. And thank you so much for accepting our invitation. So
thank you.
Okay, you can share your screen with us and feel free to start.
Thank you so much, Ana Paula, and thank you all for the kind invitation. It's really my pleasure to be here with you tonight. This is the outline of my talk. After a brief introduction on triple-negative breast cancer, I will discuss on the heterogeneity of this disease, and then I will focus on the low-grade forms of TNBC, in particular on the genotypic phenotypic correlation in triple-negative breast cancer and the new diagnostic applications. Triple-negative breast cancer is truly not one disease. We should see it as a collection of diseases. It's really so heterogeneous that it's not defined by what it has, but rather by what it lacks, which is the expression of estrogen receptor, progesterone receptor, and HER2. Triple-negative breast cancer accounts for approximately 15% of all breast cancer. When we think of TNBC, what comes to mind are these high-grade common forms of triple-negative breast cancer, which are characterized by high pleomorphism, extensive TIL infiltrate, and enrichment for genetic alterations affecting genes that are involved in homologous recombination deficiency, such as BRCA1. However, this is not all TNBC. TNBC is really vastly heterogeneous. It's heterogeneous at the genetic level, transcriptomic level, and also, as I'm going to show you, histologic level. So starting with the transcriptomic heterogeneity of TNBC, there were really pioneering studies in this field about a decade ago that were led by Lehman et al. And Bernstein et al. And these authors proposed this classification of triple-negative breast cancer based on gene expression in 4 subgroups. 2 groups that are the basal-like groups, basal-like immunoactivated and basal-like immunosuppressed, or BLIA and BLIS. We have also the mesenchymal subgroup and a very distinctive group, which is called the luminal androgen receptor, or LAR. So the basal-like subgroups of TNBC are characterized by complex genomes, high frequency of homologous recombination deficiency, and very frequent mutations affecting TP53. BLIA and BLIS differ by their TIL infiltrate. While BLIS, or basal-like immunosuppressed, have a low immune infiltrate, the basal-like immunoactivated tumors are characterized by a rich TIL infiltrate. Now we have the mesenchymal subgroup that is have a much lower genomic complexity than the basal-like immunoactivated tumors, and characteristically shows activation of the PI3 kinase pathway. And finally, we have the luminal androgen receptor tumors, which are very distinctive. They have low genomic complexity, they have increased expression of androgen receptor, and they frequently harbor mutations in PIK3CA, AKT, and GATA3. But this classification of tumors is not merely an academic exercise. This has important clinical implications, as these different subgroups of triple-negative breast cancer have different responses, for instance, to neoadjuvant chemotherapy. So in contrast to basal-like immunoactivated tumors that have high rates of pathologic complete response following neoadjuvant chemo, we have the luminal androgen receptors, or the basal-like immunosuppressed tumors that have very low rate of PCR. Triple-negative breast cancer is also quite heterogeneous at the genetic level. In general, triple-negative breast cancer is characterized by complex copy number profiles. They have frequent amplifications affecting MYC, frequent homozygous deletions in important tumor suppressor genes, such as PTEN and CDKN2A. In the mutational landscape, they have recurrent mutations in TP53 and PIK3CA, but they don't have really a characteristic genetic alteration. But we have to take into consideration that the large-scale genetic efforts to characterize triple-negative breast cancers have been focusing on common forms of this disease in the garden variety high-grade triple-negative breast cancers. However, triple-negative breast cancers have high-grade tumors and also low-grade tumors. And the identification of these low-grade variants of triple-negative breast cancers is really important, because not only they are different phenotypically, that they have different morphologies that are very distinctive, they have also different genetic alterations, they have a different biology. They oftentimes are underpinned by pathognomonic or disease-defining genetic alterations. And to be cognizant of their existence is really key, because these tumors, in contrast to common forms of triple-negative breast cancer, have an indolent behavior, which has important therapeutic and clinical implications. A couple of years ago, we proposed the following molecular taxonomy for triple-negative breast cancer based on their evolutionary pathways. On one hand, we have the conventional high-grade triple-negative breast cancer, which is characterized by high genetic instability and complex copy number profiles. We also have a second group of triple-negative breast cancer, which we can coin as the low-grade triple-negative breast neoplasia family. This has different members, including microglandular adenosis, atypical microglandular adenosis, and actinic cell carcinoma. These tumors have a very indolent behavior. They have a bland morphology. Nonetheless, they share with the conventional high-grade triple-negative breast cancer aggressive genetic features. They're also very genetically unstable. They have complex copy number profiles. They have also a high frequency of genetic alterations affecting the PI3 kinase pathway and frequent PIK3CA mutations. And finally, we have this fascinating group of entities, which we can group as salivary gland-like tumors of the breast. These are tumors that are morphologically indistinguishable in the breast and in the salivary gland. And oftentimes, these tumors are underpinned by pathognomonic or disease-defining genetic alterations. For example, one of these tumors is the secretory carcinoma that we know is characterized by the ETV6 ANTRAC3 fusion gene. And this is important not only academically and diagnostically, but also therapeutically, because nowadays we know that the ETV6 ANTRAC3 fusion gene can be targeted with TRK inhibitors. And actually, secretory carcinomas of the breast, even in the metastatic setting, have shown durable and really long-lasting responses to TRK inhibitors. Hence, the dissection of the biology of these low-grade variants of triple-negative breast cancers is really important. So in the second part of this talk, I will focus on this fascinating group of entities. One of these salivary gland-like tumors of the breast is adenocystic carcinoma. We have the classic variant, and for the pathologist, I don't need to describe this, but for the rest of the audience, I can tell you that this is a tumor that can be diagnosed without a microscope, has really a very peculiar morphology. This is a biphasic lesion, has epithelial and myoepithelial component, has lumina that are lined by epithelium and also pseudolumina that are lined by myoepithelium that contain this basement-like material. These tumors, when they arise in the breast, are histologically identical to adenocystic carcinomas arising in the salivary gland or in other anatomic locations. Importantly, they have an indolent behavior, so they have to be distinguished from the high-grade triple-negative breast cancer of the breast. Most of these tumors are driven by the translocation 6-9 that results in the MIB and FIB fusion gene, which would subsequently result in activation of the MIB signaling pathway. Notably, we conducted a study analyzing the adenocystic carcinomas of the breast that lack MIB and FIB fusion gene. And we observed that these tumors were characterized by rearrangements affecting not MIB, but a paralog of MIB, which is called MIBL1, or by amplifications of MIB, suggesting that breast adenocystic carcinoma is really an example of a genotypic phenotypic correlation arising in the breast and of a convergent phenotype, where we have a activation of the MIB signaling pathway by different molecular mechanisms. But there is another type of adenocystic carcinoma of the breast, which is the solid basaloid adenocystic carcinoma, or ADCC. These tumors are different, they are rare, they are characterized by a predominance of epithelium. Histologically, they have also a distinctive phenotype. They have this geographic pattern of growth, or they're composed by islands of basaloid appearing cells. The stroma, which is the intervening areas between the tumor cells, is frequently myxoid or hyalinized. And in contrast to the classic forms of basaloid adenocystic carcinomas, which are very mild and have mild atypia, these solid basaloid ADCCs are characterized by moderate or marked atypia and brisk mitotic activity. So we sought to determine what is the genetic underpinning of these tumors. We accrued a cohort of solid basaloid adenocystic carcinomas and of classic adenocystic carcinomas, and observed that in contrast to the classic adenocystic carcinomas that in most cases harbor fusion genes, MIB and FIB fusion genes, or rearrangements involving MIB or other MIB paralogs, solid basaloid adenocystic carcinoma in most cases lacked rearrangements involving MIB or MIBL1. So they're genetically different. We also assessed their repertoire of genetic alterations and observed, I'm not showing this here, this is unpublished, that they have a high frequency of genetic alterations affecting genes in the Notch signaling pathway. And importantly, the biology and the outcome of these patients with these tumors differs dramatically. So here in this Kaplan-Meier curve, we can see that in contrast to the classic, or compared to the classic forms of adenocystic carcinoma, we have in blue the recurrence-free survival of the solid basaloid adenocystic carcinoma that is much worse than the classic form of ADCC. So it's important to be cognizant of the existence of this entity. Taken together, for this part of the talk, I would like to conclude that classic adenocystic carcinomas harbor the MIB and FIB fusion gene in most cases, that breast adenocystic carcinomas, the classic type, that lack MIB and FIB fusion gene, harbor MIBL1 rearrangements or MIB amplification, and that solid basaloid adenocystic carcinomas are different tumors that in most cases lack rearrangements affecting MIB or MIBL1 and have a worse outcome and a shorter recurrence-free survival than classic adenocystic carcinomas. Now I would like to touch on other very interesting entities arising in the breast, which are the pleomorphic adenomas. So these tumors, breast pleomorphic adenomas, are vanishingly rare. They're so rare that many authors question their existence. Breast pleomorphic adenomas are well-circumscribed lesions that are characterized by the admixture of epithelial and myoepithelial components that are forming glands or clusters that are immersed in a myxochondroid stroma. Pleomorphic adenomas, when arising in the breast, as in the other locations, have usually a very benign course, although in the breast, there have been reports on local recurrences. given their rarity, the diagnosis of pleomorphic adenomas is very challenging. And because of how rare these entities are, and because of their mixochondrial stroma, oftentimes the distinction from pleomorphic adenomas that have a very benign course from metaplastic carcinomas that are aggressive tumors is quite challenging. So we conducted this study where we subjected pleomorphic adenomas to RNA sequencing. And we observed that pleomorphic adenomas in the breast also harbor HMGA2 and WIF1 fusion genes, and beta-catenin and PLAG1 fusion genes, which are the fusion genes that characterize pleomorphic adenomas in the salivary gland and in other locations, showing that first of all, pleomorphic adenomas in the breast do exist, and they have a genetic underpinning that is similar to that of their counterparts in the salivary glands and in other anatomic locations. Mucoepidermal carcinomas of the breast are also other salivary gland-like tumors that are vanishingly rare. Histologically, they resemble the mucoepidermal carcinomas of the salivary glands or of other anatomic locations. They are composed of an admixture of squamous cells, mucinous cells, and intermediate cells. They are extremely rare, and because of this, oftentimes they are confused with squamous cell carcinoma. It's really challenging for the diagnostic pathology to distinguish them. We conducted this study where we subjected mucoepidermal carcinomas arising in the breast to RNA sequencing and observed that they are underpinned by this fusion, which is characteristic of mucoepidermal carcinomas in other locations, such as the salivary gland, which is the CRTC1 MAMAL2 fusion gene, showing that for both pleomorphic adenomas and mucoepidermal carcinomas, molecular studies can be very useful for their diagnosis. To conclude with this part of the talk, I would like to say that pleomorphic adenomas and mucoepidermal carcinomas of the breast harbor genetic alterations that are similar to those described in their salivary gland counterparts, and that the molecular studies of these entities constitute a useful tool to aid in their diagnosis. I would like now to discuss with you about the adenomyepithelium of the breast. This is another rare entity in the breast. Histologically, they have these nodular growth patterns. They have different histologic architectures. We have papillary adenomyepitheliomas and also tubular adenomyepitheliomas. These are epithelial myoepithelial entities. They have an epithelial component and a myoepithelial component that here we see highlighted with this immunostain for P63, a myoepithelial marker. Adenomyepitheliomas are mostly benign, but they may progress to high-grade tumors. When they are associated to a carcinoma, these carcinomas are usually triple negative, and oftentimes they are metaplastic. And in rare occasions, adenomyepitheliomas of the breast may even metastasize. We conducted this study to determine the genetic alterations affecting adenomyepitheliomas on the breast, and we accrued a significant cohort of AMEs, or adenomyepitheliomas, that were either ER negative or ER positive. So these are the results of our whole exome sequencing study. We see in this heat map the cases in columns and the genes in rows, and the mutations are color-coded according to the legend in the right-hand side. In ER-positive adenomyepitheliomas, we observed that most of them were underpinned by genetic alterations affecting genes of the PI3 kinase pathway, such as AKT or PIK3CA. Now, the findings in ER-negative adenomyepitheliomas were quite interesting, quite striking. We observed that about 60% of ER-negative adenomyepitheliomas were underpinned by HRAS hotspot mutations, and these were affecting a specific codon, which is Q61. And in all cases that harbored HRAS Q61 mutations, we also observed coexistent PIK3CA or PIK3R1 genetic alterations. These findings are notable because they suggest that HRAS Q61 hotspot mutations are pathognomonic for ER-negative adenomyepitheliomas in a breast-specific context. Following this discovery, we went to the lab. Because we sought to determine which is the functional impact of the co-expression of HRAS Q61R with PIK3CA mutations. And we conducted this study using a triple negative mammary cell models and conducted this three-dimensional cell culture and observed that upon forced expression of HRAS Q61R, there was an increased expression of myoepithelial markers, seen here in green. Green is representing calponin. That here marks the nuclei. Interestingly, when we force expressed HRAS Q61R in cells that had a knock-in mutation in PIK3CA, a hotspot H1047R, we observed that the expression of myoepithelial markers was even stronger, indicating that the HRAS Q61R mutations, particularly when they coexist with PIK3CA mutations, are the likely drivers of ER-negative adenomyopitheliomas. We had a follow-up study that was actually conducted together with Ana Paula, where we sought to determine whether we could use this genomic finding diagnostically. And we conducted this project where we wanted to see whether the HRAS Q61R hotspot mutations in adenomyopitheliomas could be detected by immunohistochemistry. So we performed the immunohistochemical staining using mutation-specific antibodies and observed that all of the adenomyopitheliomas that lacked mutations in HRAS Q61R were negative by immunohistochemistry, while 5 out of the 7 adenomyopitheliomas that had mutations in HRAS Q61R showed diffuse and strong positivity with our mutation-specific antibodies, indicating that the immunohistochemical detection of these mutations is highly specific and moderately sensitive and could be used diagnostically. In conclusion, I have shown you that the genetic underpinning of adenomyopitheliomas of the breast varies according to their estrogen receptor status, that ER-negative adenomyopitheliomas of the breast harbor recurrent HRAS Q61R hotspot mutations, which in most cases coexist with mutations affecting genes of the PI3 kinase pathway, and that the immunohistochemical analysis of RAS Q61R is specific and moderately sensitive for the detection of adenomyopitheliomas harboring these mutations. Finally, I would like to discuss this entity that's very dear and near to my heart and is truly fascinating, which is called the tall cell carcinoma with reverse polarity. This is a very interesting entity that is now included in the latest WHO classification. These tumors are very, very distinctive. They grow as invasive nodules. They have this papillary and follicular architecture, and they're composed of cells that are columnar and display reverse polarity. Reverse polarity meaning an apical location of the nuclei. These tumors go by different names. They are also called solid papillary carcinoma with reverse polarity. Another name that they have is the breast tumor resembling tall cell variants of the papillary thyroid carcinoma. And they have this tumor because histologically they do look like PTCs or papillary thyroid carcinoma. They have this very characteristic nuclear clearing and grooves and even inclusions. These tumors are vanishingly rare. There are less than 50 cases reported in the literature. All of the cases reported so far have been HER2 negative. Half of them are also ER negative, and the second half can express estrogen receptor, but usually very focally and weakly. And importantly, these tumors, despite being invasive, they have a very indolent behavior. A couple of years ago, in collaboration with the Harvard group, we conducted this study where we subjected 13 of these tumors to next generation sequencing and observed that most of these tumors harbored hotspot mutations in IDH2. It was IDH2 R172 hotspot mutations. And in most of these cases, or in all of the cases, these coexisted with genetic alterations affecting genes of the PI3 kinase pathway. Interestingly, in one of these cases that lacked mutations in IDH2, we found a loss of function mutation in TET2. And why do I bring this up? Because IDH2 and TET2 are part of the same pathway that controls DNA methylation, suggesting that TCCRPs, or tall cell carcinomas with reverse polarity, are other examples not only of a genotypic phenotypic correlation, but of convergent phenotype characterized by genetic alteration that results in epigenetic defects. Following this discovery, we went back to our MCF10 cells to determine which were the effects of the expression or forced expression of IDH2 R172 in combination with knocking of PIK3CA H1047R, a hotspot mutation in PIK3CA. And besides many other findings, I want to show this particular finding, which is quite interesting. Here we did this three-dimensional cell culture, and we can see DAPI highlighting the nuclei here in blue. We have ikadirine that shows the cellular membranes, and GM130 that shows the Golgi. And if you see the arrows here, we observed that upon co-expression of IDH2 R172 mutations together with PIK3CA mutations, we observed a reversion of the polarization of the cells that we can see with a different location of the Golgi marker here, recapitulating the phenotype observed in these tumors. And we contend that this indicates that IDH2 mutations, in particular, when they coexist with PIK3CA mutations, are the likely drivers of these tumors. There are actually several human cancers that harbor genetic alterations affecting IDH1 or IDH2. And there is an interest in the use of mutation-specific antibodies in diagnostic pathology, actually in different organ systems. For instance, in the central nervous system, there have also been studies by hematopathologists and also by our colleagues here from the head and neck service. So we sought to determine whether we could use mutation-specific antibodies for IDH2 R172 to detect the mutations in these very peculiar tumors. And we conducted this study where we accrued 14 TCCRPs, or tall cell carcinomas with reverse polarity, and many others breast neoplasm, including benign neoplasms such as papillomas, and also other papillary tumors of the breast, such as solid encapsulated papillary carcinomas or solid papillary carcinomas. And we observed that in contrast to the other differential diagnosis of TCCRPs, which were all negative by immunohistochemistry, 13 out of the 14 TCCRPs that we studied were positive by immunohistochemistry using our mutation-specific antibodies, which showed a diffuse and strong reactivity, and interestingly, a granular morphology, or a granular nature of this stain, which could be related to the fact that IDH2 is a mitochondrial enzyme, indicating that the immunohistochemical detection of IDH2 R172 mutation is feasible by immunohistochemistry, and it's actually quite specific and sensitive. Taken together, these results show that TCCRPs are underpinned or characterized by IDH2 R172 hotspot mutations that frequently coexist with mutations affecting PI3 kinase pathway genes. We have seen, using our cell models, that IDH2 R172, when they coexist with mutations in PIK3CA, result in the acquisition of the reverse polarity, and that the immunohistochemical detection of IDH2 R172 hotspot mutations is a sensitive and a specific tool for the identifications of TCCRPs. In summary, to end this talk, I would like to say that salivary gland-like tumors of the breast are characterized by pathognomonic or disease-defining genetic alterations and constitute examples of genotypic phenotypic correlations. that the genetic underpinning of the vast majority of salivary gland-like tumors of the breast is similar to that of their salivary gland-like counterparts, and that the assessment of pathognomonic or disease-defining genetic alterations can be a useful tool for the diagnosis of rare histologic variants of TNBC. And to end this talk, I would like to leave you with this important message: that triple-negative breast cancer is not one disease, but it's a panoply of diseases. TNBC is very heterogeneous, and that if we embrace rather than ignore this heterogeneity, we will be able to dissect the biology of these tumors and perhaps even pave the road for the realization of the potentials of precision medicine in breast. With that, I would like to thank you all for your attention, thank my colleagues in George Rice Fillo's lab in the breast pathology department, and thank our collaborators in other institutions. Thank you, and now I'm delighted to take questions.
Thank you, Fresia.
Fresia, thank you. Thank you so much for such an interesting and excellent talk, Fresia. This is a complex field for everybody here, and I think you made a little bit easier for us.
Oh, thank you so much.
So you've shown us how the massive parallel sequencing reshaping the way we perceive triple-negative breast cancers. I'd like to know how frequent do you use the new classification for triple-negative breast cancer in the clinical setting?
Well, I think now with these studies, we're trying to find the diagnostic applications. It's not only to publish paper and write books, but we want to improve diagnosis and improve therapy. So in the practice, at least as a pathologist, I think now when we see a tumor that could look perhaps like an adenocystic carcinoma, we have to study it. And now we can conduct FISH. And if we don't have FISH, one can do MIB, like a MIB immunohistochemical stain. And actually, for example, for adenocystic carcinoma, we have observed that tumors that have MIB genetic alterations, rearrangements of MIBL1, or those that lack any genetic alterations in MIB, they are all staining for MIB immunohistochemistry, showing that these tumors are driven by increased signaling in this pathway, regardless of the molecular mechanism.
And then you can have available another type of, a specific type of treatment for them.
Right, like it's important to distinguish a classic adenocystic carcinoma that, in our opinion, does not require any chemotherapy, than from a common
from a solid type.
Exactly, from a solid
that is
type.
very hard to diagnose. I think that this kind of tumor is very hard to diagnose, and when you have the opportunity to make this MIB study, this genomic study, or the FISH, or whatever you can do to have this response, you can treat this patient with special treatment.
Right.
Give them this opportunity.
And also with the introduction of these mutation-specific antibodies, we can also facilitate the diagnosis of these very rare entities.
Yes.
Without the need of subjecting them to expensive sequencing studies. One can do a simple immunohistochemical stain. And
exactly.
if this is positive, one can confidently say,
of course, yes.
This is an adenomaepithelioma, or this is a TCCRP.
Yes, we can use this in the practice, in the practical pathology. No, so anyone would like to ask any question or comment? Karina, algum outro comentário?
Ana?
Oi.
É Gisarte, eu só não posso... Tudo
Onde
bem?
Oi, Gisarte.
Só não consigo aparecer na tela no momento.
Tá ótimo.
Eu vou falar em português, você traduz para ela?
Tá bom?
por favor.
Ela fala espanhol também, tá?
Tá, não, é só porque no momento tô, enfim,
bom.
mais fácil falar em português.
Tá.
Tiveram uns estudos na ASCO desse ano com imunoterapia que
Uhum.
começaram a demonstrar uma diferença entre os tumores triplo-negativos, a resposta à imunoterapia conforme os subtipos moleculares, como ela mostrou no começo.
Uhum.
Então, o que eu queria saber é se no Memorial, que é um hospital de ponta, se eles já fazem algum tipo de distinção pela análise molecular no tratamento, não sei se ela vai saber sobre o tratamento especificamente dos tumores triplo-negativos, e o que falta para a gente aplicar na nossa prática, né? Se ele já tem essa disponibilidade de avaliar mais a fundo o tumor triplo-negativo.
Tá, foi mais ou menos essa pergunta que eu tinha feito para ela, deixa eu fazer alguma coisa parecida. Você está querendo dizer só nos casos que vão para imunoterapia?
Sim.
Os avançados?
Enfim, nos avançados, né? Para definir terapêutica. Eu achei que você estava falando mais dos tumores raros ali.
Tá, não, vamos ver, deixa eu perguntar para ela. Fresia?
Yes.
We have a oncologist, a clinical oncologist, that she said that in the last ASCO meeting, they comment something about treatments that can help in the immunotherapy decision, and even new another treatments that could be done if you could separate or classify even the high-grade triple-negative breast cancers. So in your institution, do you frequently study the triple-negative breast cancer high grades when they are advanced?
Yes, thank you. That's
trying
an excellent
subtyping them.
Isso.
Right. Well, I can tell you what we can do here regarding tools to triage patients for immunotherapy. To how can we identify patients that can benefit from immunotherapy? So first of all, when I get a biopsy and it's a triple-negative breast cancer, the first thing I do is to comment on the TILs. So I would comment if the case have like important host lymphocytic response, because perhaps later this would help triage this patient for immunotherapy. And another thing that we are doing more increasingly, almost routinely, is the assessment of PD-L1 in triple-negative breast cancer. So we do this with the antibody with SP142. That's the clone that we used. And these cases that are positive for PD-L1, and positivity is defined as positivity in more than 1% of the immune infiltrate in the tumoral area, have been shown benefit by immunotherapy. And one last thing that I would like to comment regarding immunotherapy is on a study that we have recently conducted on Lynch syndrome patients. This is not yet published. And because we know that microsatellite instability, it's important, tumors that are microsatellite unstable may benefit from immunotherapy. So we have observed that patients with Lynch syndrome that have breast cancer, in those cases, oftentimes the breast cancer does show microsatellite instability. They're very rare, but they do exist. And in anecdotal cases in our cohort, we have seen outstanding response to immunotherapy in these cases. Thank you for the question. It was truly excellent.
Okay, thank you.
Fresia, you talked something about the microsatellite instability, and I have a curiosity because in the last edition of WHO classification of breast tumors, they did not include Lynch syndrome.
Right.
So I think it's, well, I don't know. I think that because the microsatellite instability is very rare, but even though I don't know why they didn't include, can you comment something about this?
Maybe perhaps we didn't publish our study on time.
Oh,
so
good.
In the next edition, it will be included.
Yes, probably.
And
You're
another reason
right.
is because it's extremely rare in breast.
Yes, because it is extremely rare, but the syndromes, they are always rare or extremely rare. And if you have a patient that has the syndrome, this is, I mean, 100% of the problem, you know. So,
Right.
you know what I mean?
Yes.
So it's something that I really sound strange for me that they take out they didn't include Lynch syndrome.
Right. So breast cancer in Lynch syndrome patients is not as common as colon cancer or endometrial cancer.
Exactly.
And that is reflected, and this is also reflected by the fact that we do not routinely assess expression of MMR proteins, right? Like it is done in gynecology or by our colleagues in the GI service for colon cancers. But as you pointed out, having an adequate clinical history indicated that a patient has a Lynch syndrome, it's a very good clue for us to work up these tumors. Not all of the breast cancers arising in Lynch syndrome patients are microsatellite unstable, but a good % of them are.
Okay.
Ana Paula, I have a question.
Lu, fique à vontade.
Okay. Fresia, thank you so much for your talk. It was really outstanding. I have a question. Do you see in the cases, in your cases, any correlation with this diversity in terms of pathology and molecular profiling associated with race? I mean, we know that African-American patients, for instance, their triple-negative breast cancer subtypes are different from the Caucasians and so on. So I wonder if you see any of this diversity according to race in the cases in your institution.
That's an excellent question. I'm afraid to say that the study would be very different to conduct given the rarity of these low-grade variants. In many studies that we conduct in these very rare low-grade variants, we are happy if we find 6 of these cases, if we find 10 of these cases. But of course, as you pointed out, it might be interesting to see whether there are race factors in here. That would require, however, multi-institutional efforts to be able to collect a larger number of cases. But yeah, that's a very, very interesting question.
Yeah, I don't know how diverse is your population in the institution, but I assume it's mostly Caucasians, right?
No, we have Caucasian people, and it's reflective of the population in New York that is quite diverse, and we have also a large number of international patients.
Oh, okay. Thank you.
Thank you.
Ok, Karina, acabaram as perguntas?
Ninguém mais tem pergunta? Acho que não, Ana.
Ok, Fresia, no more questions for you tonight. And
thank you so much.
I'd like to thank you. Thank you for your kindness. Thank you for participating with us tonight. And I hope to see you soon in person again.
Me too.
As soon as possible.
Me too. Thank you so much. Thank you all for joining. Thank you, Ana Paula, for this kind invitation. It's for me a real pleasure to be talking to you tonight. Thank you for these very, very interesting questions. And I hope you invite me again and I can tell you more about our ongoing studies.
Sure, absolutely. Thank you, Fresia.
Thank you all. Have a good evening.
Good evening.
Thank you, thank you.
Bye-bye to everybody.
Bye-bye, boa noite a todos. Semana que vem temos Tumor Board na nossa reunião, tá bom? Thank you, Fresia. Thank you for staying with us tonight.
It's my pleasure. Thank you.
Eu gritaria